Hello students, our today's topic is DNA fingerprinting, which is a part of molecular biology. Before start with DNA fingerprinting, first of all we should know that what is forensic science, because this technique is very useful in forensic science. Forensic science is an insertion of law and science. Here the science as well as law both are amalgamated with each other. Historical example, first of all, in 1800, this technique, not actually this technique, such kind of technique, photography, was evolved to search the criminals and all. But in early 90s, fingerprinting was discovered, where the offenses or criminals were arrested on the basis of their fingerprints. Now, the actual DNA fingerprinting technique was discovered in 1985, and this is actually the unique signature found in each person's genetic makeup. Every person has different fingerprints, so we can easily detect any criminal on the basis of his or her fingerprints. Now, DNA fingerprinting is a technique which is used by the scientist to distinguish between individuals of the same species using only a sample of their DNA. We can use this technique for comparing the various species or for comparing the similar species. So we are all nearly unique. Most DNA is highly conservative from one person to the next, but a small domain 0.1% are very variable. Uh, there are some historical examples. In 1800, photography was used. In early 1900, fingerprints or palm prints was used to detection of the same species or the criminals. Then retinal scans, we have footwear and their tire impressions, other ear lip impression, etc. These were the earlier techniques which we were using for matching purpose. And now DNA fingerprinting, I have already told you that it was discovered in 1984 and 85. Now, what is actually DNA fingerprinting? Now we are having the definition. DNA fingerprinting is a forensic technique used to identify individual by characteristics of their DNA. The process of DNA fingerprinting was invented by Alec Jeffrey in 1985 at the University of Leicester. It is also called as DNA profiling or molecular fingerprinting. Only 1/10 of 1% of DNA will differ from person to person, otherwise the DNA is same. And that 1/10 of 1% of DNA makes us unique. Why we are using DNA fingerprinting? So DNA fingerprinting is a way of telling individuals of the same species apart. DNA sequences are variable and can therefore be used as identifying characteristics. DNA fingerprints has advantage over other sources of evidence. Fingerprints, blood type, etc. They are highly accurate, can be gathered from the trace crime scene evidences, and it is a process or a technique of analysis revealing unique pattern of an individual's DNA involving non-coding region. This we know very well. In DNA we have two types of region, coding sequences as well as non-coding sequences. So by the help of this technique we can also scan, or we can also amplify the non-coding regions. Now, DNA fingerprinting and profiling, in which the genome in a particular species is more or less same in every member of a species, but the genome contains polymorphisms, positions where the nucleotide sequences are not same in every member of the population. So in 1980, American researchers discovered non-coding regions of DNA, but in 1984, Professor Alec Jeffrey, you can see here the pictures of Professor Alec Jeffries developed this DNA profiling. So he was the first to comprehensively research on DNA fingerprinting. In 1987, first conviction based on DNA evidences was done. Now, types of DNA fingerprint testing. There are two ways of forensic DNA testing. One is RFLP, in which we are talking about restriction fragment length polymorphism, and another one is polymerase chain reaction PCR. In RFLP, it requires large amount of DNA, and DNA cannot be degraded. In PCR, we required less amount of DNA, and DNA can be partially degraded, and extremely sensitive to contaminating DNA. So here PCR is more useful uh, as compared to the RFLP for testing DNA, because it requires less DNA and it can be partially degradable. DNA profiling depends on a small portion of the genome, exon code proteins, as well as the intron, which do not code for proteins. The intron contain repeated sequences of between 1 to 100 base pairs, and these are called variable number tandem repeats, VNTRs. Some VNTRs are inherited from mother and some are from father. Now, in this diagram, you can see there are two set of DNA, two set of small fragments of DNA, in which red color shows repeated sequences of VNTR locus. Now we run both the DNA samples, maternal side and paternal side. We will found that in some paternal side and parental side, and second one, we have some samples, sample A, sample B and sample C, and one is the forensic sample, which is the 3 pair of homologous chromosomes. Now we can see here in this diagram, then from father side, from a forensic sample resembles more, and then we compare the forensic sample with A, with B and with C. The number of VNTR repeats, if they are same with the F sample, that means that is the original individual, and that individual was from that particular sample. Now, biological samples which are used for DNA fingerprintings are what kind of samples we can use for this process. The first one is blood, then we can take hair, saliva, semen, body tissue cells. DNA sample have been obtained from vaginal cells transferred to the outside of a condom during sexual intercourse. Now, the methodology. There are 5 steps which are involve in DNA fingerprinting. The first one is DNA extraction, then DNA cutting, gel electrophoresis, southern hybridization, and autoradiography. Sorry, it's autoradiography. The first step is DNA fingerprinting, in which the isolation of DNA must be done, and DNA must be recovered from cells or tissue. Only a small amount of blood, hair or skin is needed to isolate DNA. Here you can see in these appentrophs DNA has been isolated. Now the second step is what the isolated DNA should be digested by restriction enzymes and specially restriction endonucleases. The DNA is cut into fragments using restriction enzymes, and each restriction enzyme cut the DNA at specific base sequences. As you can see here, the first cut generated between G and A, and second one is between A and G. So here whatever the segments or the DNA segments we have get, those are having sticky ends. The section of DNA that are cut out are called restriction fragments. These yields thousands of restriction fragments of all different sizes, because the base sequences being cut may be far apart, long fragment, or close together, short fragment. Commonly used restriction enzymes are HAE3, HINF1, ALU1, etc. The action mixture is incubated overnight at 37 degrees centigrade. Now, the third step is electrophoresis, in which separation of different fragments are separated on the basis of their size using a process called gel electrophoresis. DNA fragments are injected into well, and electric current is applied along the gel. DNA is negatively charged, so it is attracted to the positive end of the gel. The shorter DNA fragment move faster than the longer fragment, so DNA is separated on the basis of their size. As you can see in this picture, in the agarose gel, DNA fragments were obtained in which smaller DNA is in front of agarose gel, or the larger DNA is in backside. Now the step 4, that we transfer DNA on nylon or nitrocellulose membrane. Now whatever the DNA fragments we are getting on the gel, we should know okay, where is the DNA, because we cannot see the DNA without staining it. So we need to transfer it on a nitrocellulose membrane or a nylon, and then we will stain it so that we can so that that particular DNA segment get the color, and we can identify, and we can actually analyze where is the DNA fragments. So the DNA fragments are transferred to a nylon sheet by placing the sheet on the gel and soaking them overnight by the process of southern blot. Then probing and probe labeling, adding radioactive or colored probe to the nylon sheet which is complementary to the target sequences. Each probe only stick to one or two specific places on the sheet. Now, the hybridization labeled probe DNA should be hybridized with complementary sequences located on nylon membrane for the detection of position of later. Membrane is washed to remove non-specific binding and clearing the background. As you can see here in this diagram, we can clearly see the bands of DNA. Now, the next one is autoradiography, which is used to detect the sequences in genome bound with hybridized radioactive probe on membrane. This technique involves alignment of hybridized membrane with X-ray films. The X-ray film alters its development, shows multiple number of bands that look like barcode and known as DNA fingerprints. Now, the interpretation of band pattern, analysis of band pattern of different individuals, comparison of position of bands. Computer software are also available for the analysis of DNA fingerprinting. Now, the uses of DNA fingerprinting, diagnosis of inherited disease disorders, hence diagnosed disorders in both paternal and newborn babies. Disorders may include cystic fibrosis, hemophilia, Huntington disease, familial Alzheimer's, sickle cell anemia, thalassemia, and much more. Crime, forensic science is the use of scientific knowledge in legal situation. The DNA profile of each individual is highly specific. The chances of two people having exactly the same DNA profile is 30 million to 1, except of identical twins. DNA fingerprinting can solve crimes. The pattern of the DNA profile is then compared with those of victims and the suspects. If the profile matches, the suspect is provides strong evidence that the suspect was present at the crime site. If the profile does not match the suspect, then that suspect may be eliminated from the inquiry. Example, a violent murder occurred. The forensic team received a blood sample from crime scene. They prepared DNA profile of the blood sample, and the victim and suspects are follows. As you can see in the diagram, in center there is a blood sample or the DNA segments from crime scene. uh, Last one is the victim profile, and the first one is suspect profile. As you can see, the suspect profile is totally identical to the blood sample from crime scene. That means that suspect was present at the crime site and he was the criminal. Solving medical problems, DNA profile can be used to determine whether a particular person is a parent of a child or not. A child paternity and maternity can be determined, and this information can be used in paternity suites, inheritance cases, immigration cases. Example of a paternity case: by comparing the DNA profile of a mother and her child, identify DNA fragments in child which are absent from mother and must therefore have been inherited from the biological father. As you can see here again in center, this is a DNA sample of child. Then one side we have the DNA sample of mother, and another side we have the DNA sample of father. As you can see, 50% band pattern should be identical to father and 50% should be mother. So here you can see in child we have 6 bands, in which 3 bands are identical to mother and 3 bands are identical to men. That means they are the mother and father of that particular child. Uses of DNA fingerprinting, we can use it for personal identification. In the US Army services are just beginning a program where they collect DNA fingerprint from all personnel for later use in case they need to identify casualties and missing people. Some other uses are identification of car cases of tissues, detection of somatic mutation or cancer, pathogen identification, detection of loci controlling quantitative traits or disease resistance, sex determination, and individual identification. These are the famous cases uh, which were detected on the basis of DNA fingerprinting. In 2002, Elizabeth Hurley used DNA fingerprinting to prove that Steve Bing was the father of her child, Demain. Colin Pitchfork was the first criminal caught based on DNA fingerprinting evidences. He was arrested in 1986 for the rape and murder of two girls and was sentenced in 1988. Thank you. I hope I have cleared you very much about the DNA fingerprinting.